So we can see a summary of those steps that we've gone through in the lipogenesis process. So each lipogenesis turn or cycle consists of these 4 steps. So in the first step, we combine our acetyl ACP and our manolal ACP, releasing one of the ACPs and also the carbon dioxide off the manolal ACP. We're able to create this 4-carbon unit that we call aceto-acetyl ACP. So the aceto-acetyl ACP is the component that we're going to modify in steps 2 to 4 to eventually get to our butyryl ACP, where we have the carbons connected by single carbon-carbon bonds. So basically, we start with the acetyl ACP, which contains our ketone group, where we have the carbon double bonded oxygen, our carbonyl group, and then we've got that attached to 2 other carbons. So through a hydrogenation reaction, which is one of our redox reactions, we're actually adding hydrogens from the coenzyme NADPH onto this aceto-acetyl ACP, and by doing that we actually convert our ketone group into an alcohol, where we have extra hydrogens being added onto that oxygen and also onto the carbon below. So after we've formed our alcohol group, we can then release some of these atoms to form a water. So it's actually the OH that forms the alcohol group and one of the hydrogens attached to the carbon next to it, and we've done this dehydration reaction where we've now created a alkene where we have this double carbon-carbon bond. So in the top here we have our ketone. We then we turn the ketone to a secondary alcohol via the redox reaction, where we are actually reducing this ketone group, and then we do the dehydrogenation to convert the alcohol into the alkene. And then the last step is to do another redox reaction where we take the hydrogens off our NADPH and put them onto this contonal ACP, which is able to break that double carbon-carbon bond and instead replace those bonds with extra hydrogens. So we've now got to the stage where this group that we're modifying is actually an alkane. And when we've got to this stage, we now have our butyryl ACP. So remember, our fatty acids don't just contain these 3 carbons, which have single bonds. So the butyryl ACP contains these 3 carbons at the end here that contain the single carbon-carbon bonds, and then we've got the attachments to the carbonyl group and also the acetyl carrier protein ACP. So if we want to make this longer, we actually start the next cycle of lipogenesis, but instead of starting with acetyl ACP, we instead start with the butyryl ACP. And by adding that to a manolal ACP, we're able to build onto the butyryl ACP an additional 2 hydrogens, just like we did in this reaction. So essentially, the end product of lipogenesis can be used to start the whole reaction again to put on another 2 carbons. So every time we do lipogenesis, we're creating these 2 extra carbons that contain single carbon-carbon bonds. And eventually we're going to get to the stage where we have lots of single carbon-carbon bonds formed between carbons, which is what we see in our fatty acid chain when the fatty acid is saturated. So the cycle basically just repeats until we've got our fatty acid with enough carbons attached. So just a reminder that the important part of this process is the knowledge of the interchange between the functional groups. So going from the ketone to the secondary alcohol to the alkene and then to the alkane. And it's also important that you know what types of reactions actually facilitate this. For example, to go from the ketone to the alcohol, we need to be reducing the ketone through a redox reaction. Whereas to go from the alcohol to the alkene, we need to do a dehydration by removing water. So what is the actual outcome of a lipogenesis cycle? So basically it's the point shown here in red. We've actually added 2 carbons onto the fatty acid chain that contain single carbon-carbon bonds. So remember, at the beginning of the reaction, we started with our acetyl ACP. So acetyl ACP had a CH3, C double bond O, and then we attach that to the ACP. So by going through this whole reaction, essentially what we've done is we've added another 2 carbons onto this chain. So we've extended the acetyl ACP number of carbons by 2, so we've plus 2. So every time we do a lipogenesis cycle, we can add on another 2 carbons by undergoing all these reactions. So essentially that's the purpose of lipogenesis, to create these additional 2 carbons which are formed, which are attached by single carbon-carbon bonds to eventually get to our fatty acid, which contains single carbon-carbon bonds, and then we've got our carboxylic acid group at the end here. So basically we can say that the lipogenesis cycle will lengthen the saturated fatty acid chain. So we're just adding these additional carbons which are connected by single carbon-carbon bonds to make the fatty acid chain longer. Because remember, we need these fatty acids to create our triacyl glycerols and store that as fat. So to make our triacyl glycerol, we have the glycerol and we attach that glycerol to 3 different fatty acids. So through this lipogenesis cycle, we're actually able to build these fatty acids 2 carbons at a time through all the steps that we have discussed. So basically, the more cycles of lipogenesis that you do, the more carbons you're going to have on your fatty acid. So you can see the summary down here of what we've actually done in this first cycle of lipogenesis. We have the derivative of the manolal ACP here, which is actually our acetyl ACP with 1 less hydrogen on the carbon here. And we've also added on these 2 extra carbons by modifying the group that we've added. So basically we've modified that aceto-acetyl ACP to now form the butyryl ACP, which contains these 2 extra carbons that contain only single carbon-carbon bonds. So in the next step, we're going to add an additional group to modify here, and then we'll change that into another 2 carbon compound, which has these single carbon-carbon bonds. Alright, so each lipogenesis cycle is setting us up for the next cycle where we can add another 2 carbons. So for example, we saw that first lipogenesis cycle produced a 4 carbon compound, the butyryl ACP. So if we do the next cycle, then we increase it by 2, so we get up to 6, and the next cycle after that would be 8, and then 10, and so on. So this elongation of the fatty acid usually stops when we reach 16 carbons, and we form what we call palmitic acid. So to produce longer saturated fatty acids and unsaturated fatty acids, we actually require different enzymes and have to go to different locations in the cell. So we're not going to talk about that today. So to create these fatty acids actually requires quite a large amount of energy input. So, because what we're creating is these energy-rich fatty acids that contain these single carbon-carbon bonds. So it's an energy-using process to do our lipogenesis. So to create our palmitic acid that contains 16 carbons, no double bonds, which we see by the 0 here, then essentially what you need is you need 8 acetyl-CoA, you need to put in 7 ATP, 14 NADPH, because remember we need 1 for each of those redox reactions in lipogenesis, and we also need some extra H plus to combine with the H plus that's coming off our NADPH. And this will give you the 16 carbon fatty acid, which we can call palmitic acid or palmitate. You'll also be releasing 8 CoAs, 7 NADP, so that's coming from the ATP after it's released phosphate. So we've got the phosphate and the ADP separated after breaking the high energy phosphoanhydride bonds that links the ATP together. That's how we get the energy released. We'll also have 14 of the oxidized NADP plus, and we'll also have produced water in those dehydration reactions. So the key thing that we're putting in is the acetyl-CoA. It's energy requiring, and we also need the NADPH coenzyme, and essentially what you get out is the fatty acid, and we've modified our other groups, so the ATP has lost a phosphate and the NADPH has lost its H plus and electrons. Alright, so we can see a summary of how we actually build up this 16-chain fatty acid. So essentially, we combine the acetyl ACP with the manol ACP at the beginning, and together they will give us the acetyl ACP. So that's what we get from our first condensation reaction. And then through the rest of the lipogenesis phase, we're able to turn our acetyl ACP into the butyryl ACP that contains those additional 2 single carbon-carbon bond containing carbons. So that's our first product of lipogenesis. We can then put that back into another cycle of lipogenesis by adding a manol ACP onto that, and by doing this we're actually able to get another one of the ketone groups that we can actually modify and then turn into another 2 carbons here that contain only single carbon-carbon bonds. So essentially what the manol ACP is giving us is this ketone group, which we can turn into the alkane group, where it just contains the single carbon-carbon bonds. So when we've finished one lipogenesis cycle, we just start the next one by adding the previous product, for example here the C6 fatty acid, with the next manol ACP, and we just keep repeating that cycle to add 2 carbons at a time. So eventually we'll get to the stage where we have the C16 fatty acid, and that's where the cycle will stop. So how do we actually create a 16 carbon fatty acid from our original acetyl-CoA? So basically we need to undergo many cycles of lipogenesis where we can add carbons that contain single carbon-carbon bonds, 2 at a time. So what groups do we use to create our initial product of lipogenesis, which is our butyryl ACP that contains the 4 carbons shown here? So basically we need our inputs as the acetyl ACP and also the manol ACP, which will combine together to form this acetyl ACP, which can be modified to our 4-carbon compound, which contains these additional 2 carbons that contain single carbon-carbon bonds. So once we have a 4-carbon compound, what do we have to do to add another 2 carbons that have the single carbon-carbon bonds onto that? So basically we have to put our 4-carbon fatty acid back into another cycle of lipogenesis. So we add a manol ACP to that 4-carbon fatty acid, and through undergoing another cycle of lipogenesis, we're actually able to generate a fatty acid which contains an extra 2 carbons. So we can see that summarized here. We're going from our C4 to our C6, so that's plus 2 carbons. And those carbons are here, and they contain single carbon-carbon bonds between them. So we basically just repeat this cycle where we're adding in the previous product of lipogenesis, combining that with manol ACP, and going through the whole cycle to create these additional 2 carbons that contain single carbon-carbon bonds. So here we have a comparison of 2 of our processes we can do in our lipid metabolism: beta oxidation, which is where we're breaking down our fatty acids. So we're breaking our fatty acids, whereas the lipogenesis is actually the opposite. It's building up the fatty acids. So let's have a look at our beta oxidation first. So the site of beta oxidation is actually in the mitochondrial matrix, and we do this when there is little glucose to metabolize for energy. So instead we use our fatty acids to derive the acetyl-CoA, which can be used to start the citric acid cycle to start the process of towards creating ATP. So we actually activate beta oxidation by glucagon, because glucagon is functioning to take the glucose that remains out of the cell and back into the blood, where it can be carried to where it's required. So the cells need another source of energy, so that source of energy is our fatty acids, which we can use to create the acetyl-CoA. So we have the coenzyme A carrying that acetyl group, which can be used to start the citric acid cycle. So we have our acetyl-CoA that's produced from beta oxidation being introduced into the citric acid cycle. So we start with our fatty acid in beta oxidation, and we convert that to the final product of our acetyl-CoAs, which can each contain an acetyl group. So we do need our electron poor coenzymes or oxidized coenzymes to actually take electrons and H plus off our fatty acid to turn it into the acetyl groups, which can then be attached to CoA. So undergoing oxidation, as the name suggests, of those fatty acids, we also need to do a hydration reaction and eventually cleave off the acetyl group, which can be added to a CoA to create our acetyl-CoA. So it would be useful to revise the previous lecture to compare some of these aspects between beta oxidation and lipogenesis. So we're also making these electron-rich coenzymes, FADH2 and NADH, which can be taken to the electron transport chain to derive ATP. So lipogenesis, it doesn't occur in the mitochondrial matrix, it actually occurs in the cytosol. So that's why we had to take the acetyl-CoA out of the mitochondria and into the cytosol using the citrate-malate shuttle. So when would we do lipogenesis? Basically when we have a lot of glucose, and that glucose will be turned into firstly pyruvate and then into acetyl-CoA. And that acetyl-CoA that comes from our carbohydrate metabolism is used to begin our lipogenesis process. So this is activated by insulin, where we have the glucose inside the cell that can be turned into the acetyl-CoA, which we need to begin our lipogenesis process. So this time, the carrier that we're using isn't coenzyme A, it's actually that ACP protein, acetyl carrier protein. And the coenzyme is different also. So we have a NADPH, and this enzyme will actually be oxidized and release hydrogen. So we're actually reducing our acetyl components in this reaction to create our fatty acids, which is our ultimate outcome of the reaction. So the other steps, we need to undergo dehydration, where we're removing our water, and also a condensation, where we're combining the acetyl ACP and the manol ACP. And you're also left with this oxidized coenzyme at the end here. So you can see that a lot of the aspects of lipogenesis and beta oxidation are actually the opposite to each other. But essentially in our beta oxidation, we're breaking down the fatty acid and producing acetyl-CoA. So there'll be many of these. Whereas with the building up of the fatty acid, we're going the other way. We're actually using many acetyl-CoA to eventually produce our fatty acid.